Today is a historic turning point in human civilization. At 1546 today, NASA organized a paleontological fossil excavation team to Mars. In the foundation tunnel of a Mars base, a large number of neatly arranged human-shaped skeletal fossils were successively excavated. In order to answer some of our questions, we invited Professor Huagu, who is located at the International Astronautical Center, to remotely direct the excavation work. As soon as the host's voice fell, the screen changed, and the host's interface changed to a small window. An old man who looked elegant and easygoing, in his 60s, appeared in front of the public. "Hello, Professor Huogu," the host said. "Host, hello," Huogu said. "Professor Huogu, can you tell us about your latest research progress on the humanoid skeleton fossils excavated on Mars?" Now it can be said that the entire world, as long as there is an internet connection, is paying attention to this matter. "Okay." Huogu nodded slightly, and said, "In order to ensure the integrity of the fossil group and to obtain more information in a short period of time, we specifically excavated a human-shaped skeleton fossil. From this, we can also conclude that we humans are not alone in the universe." "Because they are Martians?" the host asked. Huogu shook his head. "No, the owners of these humanoid bones are not native Martian creatures. They come from other planets and belong to another ecosystem besides the Martian ecosystem." Was shocked, and the host's eyes were obviously wider than before. The breathing and tone of support for people could not help but become a little rushed, and said, "Then, which planet do these human skeletons come from? What is the evidence for this conclusion?" Huogudeo, although there are many kinds of life on the planet, as long as you look back to the source, you can find that they have the same ancestors along the evolutionary route in the past. Those of us who study Martian biology call this species at the source of evolution cornerstone species. This, can you make it a bit more popular? The host looked a little embarrassed. Take the Earth as an example. The early Earth environment was transformed into an oxygen environment. After that, the mainstream of the Earth's biosphere was to consume oxygen to sustain life activities. The same principle applies to Mars. The mainstream of Martian paleontology is the exoskeleton. The bones wrap the organs and support the skin and muscle tissue. Under this system, if there are Martians of the same size, it is better than normal people are 1.5 times heavier, but those human-shaped skeletons are not like that. Their skeletal structure is more similar to that of life on Earth. It turned out to be like this. The host nodded in a daze, and didn't know if he understood. "So, were these Martian fossils intelligent life?" the host asked. "I can definitely answer this question, yes," Huogu replied. "Each corpse is arranged neatly, forming a huge group of square fossils, which is very similar to the burial on our planet. From this, we speculate that they are wise and not low, at least they are in the Stone Age." Suddenly, the host's complexion condensed, and he listened very carefully for a while pressing the headset with one hand, and then asked, "Professor Huogu, there was a problem on the internet just now. The number of hits exceeded 1 billion in an instant, and it is still rising rapidly. Can we ask you about this?" "Of course," Huogu answered with a smile. "What do you think of the crossing thing? Is it possible that these humanoid bones were the ancestors of humans who crossed to Mars?" Huogu shook his head with a smile. "No, it's impossible. I just said that it is similar to Earth creatures, but I didn't say that they are consistent. The owner of the bones and Earth creatures are not the same system." So, a picture appeared on the lower right side of the screen. The bones are quite similar to human bones. One of the few obvious differences is that there is a hole in the forehead. We can see that there is a hole in the center of the forehead of the skeleton, which is not caused by the damage of the skull. According to the results of our computer's comparison of the intracranial structure, we found that the structure of this hole and the other two eye sockets are basically the same. From this we infer, this hole is an eye socket. In other words, the owner of the skeleton is a humanoid creature with 3 eyes. Like what I said just now, the keystone species, from the initial birth of Earth creatures to the present, no species has 3 eyes. In addition, the Mars excavation team did a simple atomic decay cycle measurement. The fossil bones existed for more than 200 million years. At that time, the dinosaurs were still ruling the Earth. But these bones are too similar to human beings. How can you explain this? Is this too unbelievable? The host asked unwillingly. There is nothing similar in bones. As long as you compare them carefully, you can easily find the 2 bones. Except for the forehead and eye sockets, the bones of humans and chimpanzees are also very similar. Are the appearances of humans and chimpanzees too similar to distinguish? Huogu hesitated for a moment, and then replied. Host, I do have some opinions about crossing. Please tell me. The host suddenly came to the spirit, Yu reading www.yuuganshu.com's eyes are piercing. Before that, let's assume that crossing is a true proposition. We all know that the universe is very large and very large. The observable universe alone is 93 billion light-years in diameter, and our Milky Way is also very huge, with about 100 billion stars. Then there is our solar system. If defined by the celestial planets, the average radius is 82 astronomical units. That is to say, if you use me as the unit of measurement, approximately 12.3 billion me can be filled. In addition, the positions of celestial bodies are constantly changing. Our home star is revolving around the Sun at a speed of 29.783 km per second all the time. It's hard to imagine that this kind of traversal will happen on the Earth so coincidentally, and, even more coincidentally, among many Earth creatures, humans have been selected as the objects of traversal. After the traversal is successful, it can ensure the maintenance of the most basic life activities. It's an incredible coincidence. Professor Huogu, you don't like the existence of crossing, right? The host asked. Huogu smiled irresponsibly. How could be? In probability, the probability value of things that haven't happened is never 0. Maybe someday in the future, there will be such a person. He has passed through, just like in the novel. What I want to say is, if a crossing does happen on a certain day, I am more inclined to think that it is a man-made event, an event triggered by the subjective behavior of an unknown civilization in the universe, rather than because of some accident. Professor Huogu. The change happened so suddenly that the host was caught off guard, and it took a long time to react, and he turned his head and shouted in disregard of the image. Hurry up and hit 120. Split again. This is the next purpose of the life body after calling for no results. In response to the surging forces in the darkness, the single cell stabilizes its body as much as possible so that it can maintain a relatively stable state. The split begins again. Repeating the same process as before, in this timeless darkness, the self-replication of life forms will soon reach the final stage. The feeling of unlocking the shackles emerged again, and even the life body could feel that the dark world around it was becoming different. When the separation outer membrane is completely formed, the 2 single cells begin to separate gradually, and the shackles appear, and the locks are slowly locked. The relaxed feeling is slowly disappearing, replaced by an indescribable sense of stagnation. The living body does not want to accept such things, it wants to continue to do so. Don't go. The life body calls instinctively like this. Should not have any reaction, but there are so many weird things in this world. The child body, which has not been completely separated from the mother body, actively engages with the life body. After the child body changes its shape appropriately, it becomes the force that fits the life body. So happy. What is happy? As always, it cannot answer questions. The living body turned its attention to itself, when it suddenly discovered that it could see more things. In the beginning, it can only rely on the feeling to see to a point. Then, after being attacked by that phage, what you can see extends from the dots to your entire body. Now, the vision in the dark world has been expanded again, it can see the child that is close to itself, the entire internal situation. Yes, this is a change, the same change as before. The living body feels happy about this. It doesn't understand why it wants to be happy, but it feels this way instinctively, so it obeys this instinct. I want more changes. This is desire, and the living body has learned desire without knowing it. Driven by this desire, it wants to continue to replicate itself. Self-replicating materials are not scarce, and there are everywhere in the dark. Every time a living body is bound to move by force, a living body can always bump into a bunch of such things. With the help of the copy that clung to the mother body, the living body quickly collected all the materials it needed. More and more. In the pleasant bioelectricity of the living body, a new division begins. The members who hugged each other in the dark world suddenly added 2 more, and the subbody also replicated itself under the command of the living body. What followed was the shock of unlocking the yoke twice. A lot great. Enjoying this feeling of comprehensive upgrade, the unraveling of the yoke is accompanied by more and more fresh things. Compared with the hazy feeling when it is a single cell, everything seems to be gradually become organized and clear. What follows is a greater desire, which drives the life body to act. It's just the close contact of 4 individuals to feel the current level. Then, what will happen if more replicated individuals are grouped together? The act of self-replication in the life body cannot wait. The 4 gradually split into 8, and with it emerges endless freshness, comfort after clear thinking, and countless other feelings that are difficult to express. These feelings make the living body deeply fascinated by them, and they can't stop eating them. But unfortunately, these feelings did not last long. The force of a spiral operation entraps 8 clumped cells, which are already fragile single-cell aggregates, even if they are facing the fate of disintegration. Don't. The life form resisted, but that was just a futile effort. No matter how it changed its shape, it couldn't resist the power that emerged in the darkness. Around it, once again, only one individual remained, wandering aimlessly. Fell from heaven into hell. Uncomfortable. Tried to hug the 7 subbody life forms, and once again felt the situation of a single individual, but it was even more uncomfortable and difficult. It didn't want to stay in this situation for even a quarter of an hour. The copy operation continues. Compared to before, this desire is now even stronger. When a new child appeared, while enjoying the clear thinking, the life body also realized with hindsight that the empty shell of the mild phage was still firmly attached to its outside, and it was not as good as other children. The body was thrown away by the sudden force. Why haven't you been separated? With this question, the living body began to observe the empty shell of this phage. The 6 scaffold-shaped tails composed of proteins are docked with the cell membrane to form a whole, thereby ensuring that it is difficult to separate them by external forces. What is protein? What is cell membrane again? This is the answer that the living body gets subconsciously, and it doesn't understand why it's just observing. Reading www.uuganshu.com can get such a transparent answer. Same as before, in this case it can't understand. Give up the question of no beginning and end in the pursuit, and the living body began to pay attention to collecting materials. Now that the question has been answered, there is only action left. The key point is the 6 tails with connecting function. Thanks to the special perspective, the living body can easily see the macromolecular structure of the 6 scaffold-shaped tails. Place the collected materials in the cytoplasm. The chaotic inner core began to partially untwist, a small part of experimental attempts, looking for the phage gene chain hidden in the gene chain. After so many attempts, the organism finally found the part it wanted, which is the genetic blueprint for the phage assembly tail. The gene chain began to play its role. The single chain captures the macromolecular clusters that are in a free state in the cytoplasm to complement itself. Soon, the macromolecular clusters in the cytoplasm were consumed, replaced by dozens of protein structures identical to the tails of the phage. These structures and the tail of that phage serve the same function. That is, it can form a hole with the outer membrane of the cell that it comes into contact with. The finished living body cannot wait to transport these protein structures from the cytoplasm to the outer membrane of the cell. The scaffold-like protein structure naturally protrudes from the gaps in the cell membrane and forms a hole with the replicated outer membrane that is close to its mother. Immediately afterwards, there are successively multiple protein structures. The connection between two single cells becomes more and more unbreakable. Those ubiquitous forces are no longer a threat to separate them, but instead it is a boost to make them move faster. The problem of fitting has been solved, and the number of life forms replicating the same kind has exponentially increased exponentially. 2, 4, 8, 16. The increase in the number of is also accompanied by the unraveling of a large number of yolk, and this stimulates the living body even more, prompting it to replicate and split into more similar species. As the number of replicas that clump together reaches a certain critical point, life forms have also changed in nature. I'm Huagu. The life form, that is, Huagu, now finally has a clear understanding of himself, completely separated from the previous state of haunting. At the same time, I had to accept that I became a single-cell biological cluster thinking. No, maybe I just accepted the memory of the human named Hagu. On this matter, Huagu didn't struggle for long. After all, I think and therefore I am. Only by being in the present can I think about those wild things. Right now, there are still the most difficult problems that need to be solved. That is the part of the gene that the mild bacteriophage has implanted into the DNA of this single-cell cluster. Mild bacteriophage is still a bacteriophage after all, not a good man, even if it is a mild phage. What is a bacteriophage? It is a virus that specifically infects microorganisms such as bacteria, fungi, algae, actinomycetes, or spirochetes, and is a terrorist killer in the microbial kingdom. Is just different from those toxic phages. The mild bacteriophages will have a longer reaction cycle. This is like poison, which is divided into chronic poison and strong poison. Chronic poison is not non-toxic or fatal, but the remaining period of toxicity is relatively long. What's more deadly is that the mild phage will infect the divided replicas as the single cells divide. In other words, the single-celled biological cluster that Huagu now hosts has all been infected. As long as the mild phage enters the last lysis cycle, it means that the group of single-celled organisms that Huagu hosts will also be finished. What will happen to Huagu, who loses his host carrier? The result is self-evident. Then, what should I do? Such a question posed in front of Huagu, making it fall into contemplation. Bacteria and viruses are essentially one kind of things, living organisms based on amino acids, but their survival mode is different. The gene chain is like a tape in a tape recorder. Only when connected together can it play its original role. Although it is more troublesome, this is the only feasible way. After turning back from meditation, Huagu thought of a rather dumb way through DNA unwinding, and gradually screen out the gene fragment that replicates the phage. But before that, it still needs some preparatory work. The first thing is to choose the experimental object. Huagu did not choose a single-celled organism currently in the carrier, but commanded the outermost replica to split into a daughter. After splitting out, this replica, under Huagu's command, clings to the group of single-celled organisms. This experiment started with the unwinding of the daughter's DNA. Huagu's spirit was very focused on observing the process, not daring to relax at all, for fear of missing a detail. Then there is partial transcription. The part of the unrotated recording starts from the gene fragment that replicates the protein structure, and then gradually expands and replicates to both ends. Be careful, you have to be more careful. Huagu warned himself over and over again in his heart that if the incomplete transcribed fragment forms a toxic phage, it will be a very dangerous thing for it. The first replication started. After the protein structure that can be used as a bridge is replicated, it is just some useless protein. Although it is docked with the transcribed phage tail, the shape is just a hairy spherical shell. It is completely inconsistent with the phage shell. Huigu also determined that the DNA at one end of the gene chain belonged to the single-cell cluster he is currently serving as a carrier, thus focusing more attention on the other end. The second replication started. This time it was indeed transcribed, but only the shell of the phage, and no phage gene was replicated. Huigu is not in a hurry. Those who do experiments must have enough patience. This is the basic quality of a scientific researcher. 10 years are the norm. At the beginning of the third replication, the bases of the single-strand gene spontaneously seek out their own partners to complement themselves. As the amino acids aggregate to form proteins, the phage finally takes shape. The 3 pairs of stent-like tails and the rocket-shaped protein container connected to the tail are the outer shell of the mild phage. The transcription continues. This time the amino acids are combined with each other in a completely different form to form a single chain, and independently complete the base pair combination, and enter the phage shell. Huigu knows that it has found the gene fragment that it was trying to find. However, Yu reading www.yuyuganshu.com hasn't waited for it to breathe a sigh of relief. What happened next made it nervous again, and its most worried thing appeared. The copying single strand does not stop transcribing the phage gene. The number of phage in the daughter cells is constantly increasing, and the DNA single strand is completely out of control. Genes are transcribed at all costs to make phage. Obviously, these are virulent bacteriophages. Among bacteriophages, the most threatening bacteriophage to Huigu. Huigu can probably guess what's going on. The gene duplication of the phage is still incomplete, and it is this incompleteness that caused the mild phage to be transformed into a virulent phage. According to the current development trend, these toxic phages will gradually burst the cell membrane from the inside to the outside, and then infect other single-celled organisms, repeating the behavior of infection replication explosion again and again. The first thing that bears the brunt is the single-cell cluster that Huigu hosts. Fortunately, Huigu had taken precautions against accidents in the experiment early in the morning. "Stay away from us." Huigu gave instructions to the daughter that did not form a connection with the single-cell cluster. The daughter body filled with bacteriophages immediately changed its fit state, riding on the power gushing around in the darkness, quickly moved away from Huigu and disappeared. Huigu was not idle either, taking advantage of the spewing power, quickly moved in the opposite direction. I don't know how long or how far I ran. After Huigu believed that he was indeed safe, the single-cell cluster stopped moving. The matter is far from over. So far, we are collecting information for the next step. Huigu's target is locked on the daughter of a single-cell cluster. Next, it will undergo a surgery to complete self-help. First of all, it is to untwist first, to untie the messy inner core. Soon, the single-cell DNA strands were unreservedly presented in front of Huigu. Next is intercept, cutting out that part of the gene chain expressing the phage. This is a very delicate task, especially for Huigu, who now has no hands or feet. Carefully control the enzyme's output restriction endonucleases to ensure that other parts of the gene chain are not damaged. At the same time, rely on instructions to remotely control single cells and use cytoplasmic flow to limit the endonucleases to the gene fragments that need to be excised. This kind of behavior is tantamount to writing with saliva and writing on the water. Oops! With the emergence of Huigu's chagrin, the wrong endonuclease was cut all the way, and other gene fragments were also cut. The single-cell core shrank by nearly half in a short while, until the inner exhaustion of dicer. Then, in the ligase secreted by the single cell instinctively, these fragmented gene fragments are joined to each other and once again combined into a complete gene chain. However, Huigu knew that it was no longer possible to replicate the original single cell with this greatly altered gene chain. Huigu reselected new cells in the single cell cluster as the target of the surgery. The failure just now did not deal much blow to Huigu's confidence. It never considered itself a genius, and even felt that it was a little stupid than the average person, but read a little more books than others. The operation restarted. This time, after learning the lessons from the previous one, Haga used the endonucleases in the single cells more carefully, greatly reducing the amount of endonucleases, and most of his attention was focused on the movement of the cytoplasm. Let the cytoplasm flow and guide those endonucleases to cut out the designated gene fragments. At the same time, after the cut, it just runs out. Failed. 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 Success. That's good. Just as Huogu firmly believed, it succeeded. After nearly 100 cells were lost, the gene fragment of the phage was cut out. The next step is to connect. This part is relatively simple. Huogu controls the cell remotely and uses the flow of cytoplasm to restrict the phage-expressing gene fragment to a certain area. Then, relying on ligase, the DNA that had been divided into two segments after the operation was recombined into one, forming a brand new gene chain. Began to split. Huogu commanded this, and the replica cells that had undergone successful surgery began to gradually divide into new individuals. Soon, a new individual with a single cell cluster appeared, but this new individual still showed some unexpected conditions. That is the cell wall. The replica cell is surrounded by a shell. Compared with the cell membrane, this shell is stronger and has a certain degree of elasticity, which can give the protective cell sufficient growth space. This. Why? Huogu thought for a while, not very sure. Is it the result of the incomplete phage gene expression? The operation did not completely cut the gene of the phage. Huogu only removed the part of the gene fragment that was previously known to express the toxic phage. Anyway, incomplete gene expression is also incomplete, and it is impossible to transcribe phage. For the sake of perfect removal, risking the reappearance of toxic phage? At that time, Huogu thought about it, but forget it, he is not a perfectionist, there is no need to do that kind of death. I didn't expect this to happen. The cell wall must have, because the main function of the cell wall is to protect the cell. A simple understanding is that the commoner diamond puts on the steel armor, and the defense power is not the same as before. Originally, Huogu planned to start the production of cell walls after solving the phage problem, but now Huogu fell into entanglement. Is not to say that it doesn't like such surprises, but after all, this is the cell wall obtained by accidentally. It's really hard to say whether it can meet the requirements it wants. It's decided, let's observe it for a while. Huogu made up his mind like this, his attention shifted to the other single cells and started the operation. With the first successful case, there will naturally be a second and a third time. After a while, Huogu has already practiced and made perfect, and the success rate of cutting gene fragments has stabilized at more than 80%. During this period, Huogu thought of a way to break the protein structure connection, which is to rely on division to create a new replica between two connected single cells and squeeze out the old single cells. This is somewhat similar to the principle of the ship of Theseus. Without affecting normal thinking, gradually replace those old single cells. Huogu, who thought so, did indeed do so. As the last infected single cell is replaced, the sense of crisis disappears. Reading www.uuganshu.com replaces it with an incomparable sense of peace of mind. After all, things that can threaten it are temporarily gone. Set up a good mood, Huogu did not forget his current thinking carrier, but a group of insignificant single-cell clusters, a lower level of life than the bottom plant of the food chain. Huogu thought that when it was still a human, it had at least 100 million ways to obliterate the current single-cell clusters, and when its thoughts drifted here, it shuddered. Too fragile, I still need to continue to optimize these single cells. Huogu, who established his goal, began to analyze the unexpected surprises. If the effect of these cell walls is not satisfactory, then it is necessary to undergo surgery to completely remove the phage gene fragments, and then rebuild the ones that can meet the needs. Cell wall. The first requirement is proper permeability. If the permeability is too good, the protective effect will be minimal. If the permeability is too poor, there will be less external substances, and the cells will gradually decline. The second requirement is proper flexibility. The cell wall must not be too hard or too soft. It must also have a certain degree of ductility. Too hard will limit the growth of the cell itself, and too soft will not have a buffer protection effect and cannot be stretched. In this case, the plasticity is also very poor, which is not conducive to huddling together in this environment. The third requirement is what Hwagu needs most urgently at present, and that is to be able to defend against the attacks of bacteriophages, aggressive bacteria, and fungi lurking in the dark. The first and second requirements are easy to test. You only need to place a single cell alone at the outermost periphery. The rest of the work is just to observe to see if the single cell used as the experimental object has changed, and to what extent. The real difficulty is the third. Hwagu is distressed. It is currently facing a problem. The cell wall is generated from the DNA blueprint in a single cell. If you want to narrow the tunnel of the circulating substance, you need to trim the DNA blueprint. However, Hwagu does not have the conditions to do this kind of thing now, or that there is not so much time. The next wave of phages does not know when they will encounter it, and the cell wall must be completed before then. Is there no feasible way? Hwagu asked himself like this, focusing his attention on the cell wall sample, and fell into contemplation. If the tunnel is reduced, the key to the problem should be in the tunnel. In his mind, Hwagu kept recalling the past that was still a human being, and all the memories related to the pipe were fleeting like a revolving lantern. Suddenly, it thought of something. Yes. If the channel is just reduced, it does not necessarily need to be redressed. As long as the channel wall is appropriately twisted to a certain extent, a small part of the channel will naturally shrink. The cell wall presented in front of Hwagu seemed to be gradually being disassembled and reorganized, and the drawings in its mind gradually became clear. If the channel is tortuous, it can be realized by the dislocation between the two cell walls, so that part of the channel can be reduced. Without further ado, Hwagu immediately began the construction of this new type of cell wall. As the sample, the single cell with the cell wall received Hwagu's instruction and began to unwind and record his own DNA, while Hwagu was staring at that part of the remaining gene chain of the phage. Soon, a brand new single cell with a cell wall was presented to Hwagu. At the same time, Hwagu also mastered the DNA that generates the cell wall. Directs the newly formed single cell, partially untwisting, creating a new cell wall again. The material circulation channel of this cell wall is slightly misplaced with the original cell wall, just as Hwagu planned before. With the previous experience of manipulating enzymes through the cytoplasm, the construction of the cell wall was completed in one go. Hwagu looked at the single cell with a double cell wall and was very satisfied. This single cell, two-layered cell wall is absolutely sufficient to defend against the injection of phage. The tunnel has shrunk to only a single molecular cluster. Hwagu is confident that absolutely no phage can invade this single cell through this pathway. Everything seems so perfect. This way, I can breathe a little. Hey! Hwagu, who was just about to transfer his energy to other places, noticed the abnormality. The double-walled cell used as the experimental sample is gradually losing its original activity. Within a short time, Hwagu completely lost control of this double-walled cell, which means that this double-walled cell died, and the DNA as the inner core also collapsed, and the cell membrane was no longer transparent. Transient. Why is this? Toxin? Decomposing enzymes? Antibiotic? Hwagu's thoughts flashed through countless possibilities, and they seemed to correspond to the current situation, but after thinking about it carefully, it seemed that they were not right. Calm down, there must be something wrong with the operation just now, just carefully row by row. Hwagu, who was just about to find the cause of the death of the double-walled cell, suddenly had a warning bell in his heart. The entire single-cell cluster has been in contact with a number of uninvited guests one after another, and it is the toxic phage that Hagu most unwilling to see. Hiss, more than 30 phages. No, the number is still increasing. With this amount of phages, the previous method is definitely not working, but now, the cause of death of double-walled cells has not been found. Forcible use is tantamount to death. Hopeless? Hagu seems to be able to see the ghost gate is slowly opening to it. Behind the door is the endless darkness. Do not. When the broken door will open, Lao Tzu has the final say. The approach of death aroused Hagu's anger, and the beasts were ate by it. The entire single-cell cluster seemed to be infected by Hagu's emotions, maintaining a very excited state. The excitement of single-cell clusters, in turn, acts on Hagu's cluster thinking. Hagu can really see that the flow of cytoplasm, the division of phages, everything is slowing down, and it becomes extremely clear at the same time. A single molecule can be seen in this state. Not only that, but as a human memory of the past, it rewinds and fast-forwards repeatedly. As long as it wants to, it can immediately recall every detail of that memory, just like a god. Opportunity, there is definitely a chance, but it's just a group of phages. Is like an experienced hunter crouching in the grass. All memories and knowledge are like animals passing by, unaware of the threat hidden in the grass. Suddenly, the prey appeared. I'm so stupid that I ignored such a killer. While whispering like this, Yu reads www.yukanshu.com single-cell clusters. Large areas of single cells begin to spontaneously release a large number of enzymes. These enzymes include endonuclease, exonuclease, polymerase, ligase, etc., as long as they are all manufactured. Through the flow of cytoplasm, all these miscellaneous enzymes are guided to the vicinity of the cell membrane. After the enzyme reaches the cell membrane, the membrane gradually becomes thinner, but this doesn't matter to the current Hagu. It is urgent to solve these phage. And those parasitic cells that can still be controlled, under the command of Hagu, quickly began to produce a large number of enzyme suicide, killing the invading phage genes together with itself. In a blink of an eye, the number of bacteriophages undergoing parasitism quickly dropped by more than half. Hagu did not relax because of this. It was very clear that the battle in the microbiological world was not the same as the macrobiological world, and the number often did not play a decisive role. As long as there is one phage that does not die, it can directly threaten the survival of the entire single-cell cluster. Just as Hagu watched the victory of this battle is coming, another large wave of phages landed on Hagu's single-cell cluster one after another. Damn it! Hagu cursed and directed the single cells to use their flesh to build a protective layer to isolate most of the phages. In order to maintain the number of cells, Hagu was also directing some single cells to divide. With this maintenance, the two sides entered a war of attrition. Either the number of toxic phages is too large, and the single-cell cluster led by Hagu is overwhelmed. Either the single-cell cluster on Hagu's side consumed this large group of phages. With the extension of the battle time limit, Hagu's eyes became red. Now its thinking gradually tilted in a more brutal direction. In a boiling liquid that keeps bubbling bubbles, a round creature is drifting along with the wave, riding the flow of liquid, floating in this area. Very good, everyone is doing well. Then we are going to close for the first time now, and the inner cells are ready to divide. Following Hagu's order, the openings of the floating cylinder creature in the front and back of the cylinder began to close slowly, as if a multicellular creature was closing its mouth. That's good. Hagu watched the whole process engrossly. To be honest, he still felt a little nervous. This is the first trial operation. Hagu cannot guarantee 100% that there will be no mistakes. Many things are sometimes so unexpected, just like the double-walled cells not long ago. Next, start putting enzymes into the cavity. Following the instruction, a large number of enzymes capable of breaking down proteins are produced by the epidermal cells in the inner cavity and put into the liquid in the inner cavity. The microorganisms trapped in this cavity, whether it is a virus, a bacteria, or a fungus, are all being decomposed by the spreading enzymes. The first thing to be decomposed is the microorganisms such as viruses. Their structure is too simple. The outer shell is a layer of protein, and bacteria and fungi also have a cell wall. However, the cell walls of bacteria and fungi will not last long. What they face is a group-level monster. Individual microorganisms like them want to contend with it, which is tantamount to idiotic dreams. After another period of time, the microorganisms in the inner cavity were completely decomposed by enzymes. In this solution, only amino acids were left. The inner wall of the cavity absorbs, processes these amino acids, and transmits them to the inner cells. The inner cells then deliver these nourishments to the epidermal cells as the main protection. Hugo looked at the whole process with a little relief, except for the opening and closing of the mouth at the beginning, which required it to give instructions. All the other things were done by the cells autonomously, which was considered a big improvement. After this, Hugo continued with the second and third feeding behavior. As far as the result is concerned, they are not much different from the first one, which can perfectly kill the microorganisms and decompose them. Absorbed into amino acids. Then the next step is the manufacture of the vision system. After confirming that he would not be threatened with his life for the time being, Hugo put the visual system on his agenda. Information is extremely important at all times. Hugo urgently needs to determine where he is in order to be able to formulate a survival strategy. If you want to have eyes, you need the eye point, an organelle that can respond to light. However, Hugo currently does not have such organelles. The eye point. Since I don't have it here, it's better to it from other guys. Well, just do it, capture those microorganisms, analyze their genes, and then link them to the original genes. Thinking so, Hugo started to capture the microorganisms. The cylindrical creature's mouth slowly opened. With the displacement and the flow of the liquid, the microorganisms are sucked into the inner cavity of the cylinder without resistance. When the two through channels are completely closed, a large number of enzymes are released. Just as before, a large number of microorganisms are killed by the enzymes. But compared with before, there is still a difference. After most of the virus-like microorganisms are killed, the enzymes are just consumed. Mistake? No, this is not a mistake, all this is developing as planned by Hugo. If there are too many enzymes, only amino acids are left in the inner cavity. If there are too few enzymes, the viruses cannot be completely killed, or the crisis like the previous phage may be repeated. The key is that the amount of enzymes should not be too much or too little. The previous few opening and closing of the mouth are not only for observation, but for the design of this multicellular organism, but also to know whether the virus is full of this kind. How long can it survive in the environment of enzymes? When the enzyme is consumed, the fluid in the inner cavity no longer has the function of breaking down proteins. Those protein structures that have a connection effect are stretched out, and they stand horizontally in the liquid in the inner cavity. Microbes that are trapped in the inner cavity and scurrying around can easily be captured by these protein structures. In this way, Hugo can directly observe the captured single-celled organisms from his perspective. This is a single-celled organism like a tadpole. The outermost layer is a relatively thick and loose cell wall, which only serves to reduce collisions, but cannot defend against viruses such as bacteriophages, or aggressive bacteria and fungi. Then there is the cell membrane. This one is unremarkable. The composition and structure of the cell membrane are basically the same. The last is the little tail that keeps wagging, also called flagella. This is the thing that surprises Hugo the most. You reading www.youcanshu.cm. With this sample, he can design an external organ that drives the movement of the entire single-cell cluster. In this way, the safety factor will increase a lot. At least when you encounter danger, you don't need to worry about running problems. If you put the flagella aside for now, the key is to find single-celled organisms with organelles like eye point. Hagu's attention shifted, and he began to look at another single-celled organism captured by the protein structure. This time, it is a single-celled organism with a double membrane and no cell wall. Compared with the single cell before, this single-celled organism is much more fragile. There is no flagella on the outside, but it did not disappoint Hagu, because inside this cell, there is an organelle that looks like a mitochondria. Mitochondria can be said to be the engine of the microscopic biological world. Combining nutrients and oxygen, or other combinations, can realize the conversion of energy forms. For example, light energy rotates energy, chemical energy turns light energy, chemical energy turns heat energy, and so on. Even if the original suspected mitochondrial organelle does not meet the current needs of Hagu, with this mitochondria as a template, Hagu can completely design a mitochondria that belongs exclusively to single-cell clusters. This is really good, but it's better to let it go first. Finding the eye point of the organelle is the most important thing. It is too dangerous to not observe the surrounding situation. The long protein structure oscillates in the inner cavity, like a fishing line thrown by a master fisherman on the water. Single-celled organisms were fished out by this thin line one after another. Among them, there are various types of organelles, and there are all kinds of strange things, but the only thing is that no bacteria with eye point organelles have been found.